I'm Zafer Gedik from Sabancı University. I'm the chair of this session, where we have two talks. First, our plenary speaker, and then we will have an invited talk. So the first one, the first talk will be presented by our plenary speaker, Professor Susana Huelga from Ulm University. Professor Huelga received her PhD from University of Oviedo. She was a postdoctoral researcher at Oxford, then at Imperial College. And following her positions at Oviedo and Hertfordshire universities, she moved to Ulm, where she has been doing her research studies more than a decade. Her research interests include several topics in open quantum systems, quantum information, and quantum optics. And her studies in quantum networks, biomolecules, and quantum metrology are among the most cited ones in the field. Like her contributions in quantum networks, she is also a well-known figure in quantum community network through her collaborations, especially in Europe. Today, we are happy to guest her to learn about some recent developments in quantum phenomena in biology. The title of Professor Huelga's talk is Coherent Effects in Biological Systems, from reduced models to numerically exact simulations of dynamics and spectral response of pigment protein complexes. So, please, Sana.
Many thanks. Now having problems to open. Wait a second. From the beginning. Okay. Hope you can see it.
Yes.
So many thanks for the kind introduction. And it's a pleasure to actually open, pleasure and honor to open the conference to present you group work that has been done in the group roughly around the last 10 years. So I thought that it may be instructive to have a sort of historical perspective to see how the thinking about this type of coherent effects has changed, and also to show you what are the pending and then the outstanding issues. So I've organized the talk thinking that there may be people in the audience that have never heard about this actually, so that you will get a sort of overview. So the first thing we should discuss is, because talking about quantum effects in biology may sound a little bit pretentious, right? So we'll see what sort of systems are we talking about, and choose then as a case study the light harvesting processes involving photosynthesis, and discuss the type of experiments that have been performed and led to actually the word coherent be brought to this context. Specifically, one wording that all the time accompanies any discussion is the possible presence and the possible observation of long-lived coherence. So we need to understand, what does this mean? What is long with respect to what is short? And then the core of the talk would be analyzing or putting forward a biobronic model for the type of systems that are at play, where you will see how we evolve from toy models initially to try to draw some fundamental principles, to now being able to deploy machinery developed in the last years to tackle realistic environments. So in a cartoon like this would be the broad scenario. Nature has had time also to develop an incredible architecture to be able to harvest energy from sunlight and convert it into stable forms of energy, that can then be subsequently used in order to produce food, ATP. So plant, bacteria, and algae have a photosynthetic apparatus whose essential building blocks are membrane proteins, like the ones I tried to depict, borrowed this picture from a textbook. So in green, you see a network of highly absorbing molecules that are complex with a protein scaffold. So this combination is a pigment protein complex, whose role is being able to convey photons that are being absorbed from the sun to a reaction center where charge separation will take place. So what is significant is that despite the photon absorption may be highly inefficient, while a photon is captured, and especially in low light conditions, so when it's essential for survival to keep the light in the system, then the efficiency from photon absorbed to charge separated is 100%. So we are talking about then the primary steps in the complex process of photosynthesis, and specifically the excitonic energy transfer that takes place across antenna complexes on a picosecond time scale. Now, the reaction center is a relatively well-conserved architecture across species, but depending where the photosynthetic system is placed, depending on the natural conditions, then there is a large variation of antennas that have been developed. But there is one that belongs actually to a rather, probably from the biological point of view, not that relevant bacterial organism, is the famous Fenna-Matthews-Olson complex, which is simple in biological terms, given that it only involves 8 bacteriochlorophyll, so 8 pigments embedded. So these lines you see around depict alpha helices and beta sheets that are the protein cage where these bacteriochlorophylls are placed. Right. There are 2 varieties of this complex, and the S2RE one was actually the first pigment protein complex that was analyzed completely via X-ray crystallography. So this is, if you want, a little bit the James Cummings model of quantum biology, in the sense that we have a very significant body of spectroscopical information to be able to kick off doing an analysis within the theoretical framework of open quantum systems to try to understand why is it possible and what is happening for actually having a nearly 100% efficiency energy transfer. So one thing you should keep in mind, because this will be relevant when we try to put everything together, is that we have a spectroscopy for this complex, different forms of linear spectroscopy ranging from absorption to fluorescence to different forms of Decoherence, plus the X-ray information, to actually being able, with variations, these numbers have been readjusted over the years, but let's say that we can put at least initial numbers to what would be the energies of the pigments and the corresponding strengths of the couplings, dipolar couplings, that will be triggered in the moment that there is a photon absorption, right, and then the network start to talk to each other via a dipolar dielectric coupling, right? But one important thing to keep in mind is that the numbers we are putting here really come from trying to make a fit to what is experimentally observable. Among that observations are also fluorescence line narrowing and other type of measurements that characterize the spectrum of the fluctuations. So we have some initial guesses for what is the possible spectral density. So the type of fittings indicated here, that will be our initial starting point for writing up a Hamiltonian, right, come from this combination, and normally the underpinning theoretical model involves a smooth, coarse-grained spectral density. I will elaborate on this later. But let's say that for the moment we have an initial set of parameters with combined origin that would allow us to say, okay, in the first instance, I really write down a Hamiltonian involving some site energies for pigment, and then I can also write down an interaction part where I can put some numbers for the value of this electronic coupling, right? So if you think about the network of pigments alone, right, disregarding both any possible vibrations coming from the protein scaffold together with internal molecular vibrations, right, I will consider the Hamiltonian characterizing uniquely the electronic degrees of freedom. And I will call excitons the eigenstates that will come up from diagonalizing this interacting Hamiltonian. Okay? Depending on the strength of the coupling, you can imagine that the excitonic states will be more or less delocalized over the network. In the specific case of FMO, the delocalization is actually not that large. There is two pigments, 1, 2, that are strongly coupled, right? And at most, the most delocalized excitons covers about 3 pigments. Right. So the spectroscopy of FMO was actually done in '68, as far as I recall, right? And let's say that the possible involvement of coherent effects in the transfer, right? Despite very, very seminal work already in the 20s, a little bit of wild speculation, I would say that the type of results that really triggered the attention on communities that were not working in photosynthesis, like our community, were possibly the experiments of Graham Fleming and his group, and subsequently by Greg Engel, who was a postdoc in Fleming's. Of course, these things don't come just spontaneously. This is the result of transferring all the machinery that had been developed in the infrared now to the visible, so that optical transitions can also be addressed. So to all the battery of information that we have from linear spectroscopy, now we add the one that is obtained in a different manner. A different manner that maybe given that quantum opticians dominate the audience, I think you can get the gist of it if you think of a Ramsey interferometric setup, right? These are experiments where, if you think about a common ground state, think only of a dimeric system, right? Whose interaction will yield to the formation of 2 excitons, E1 and E2, with a certain gap, right? So now think about the possibility of preparing by external irradiation a coherent superposition of excitonic states. So in a Ramsey setup, that would be applying a pi/2 pulse. So now you have a part of the experiments that creates coherence. You let it evolve during a free evolution time, and now you have a second pulse that will read out that coherence by means of yielding a population. Okay, this is what you do in a typical interferometric set. So you see that the signal that we will get, should the evolution be completely unitary, would be a sinusoidal signal, typical indication of an underpinning coherent evolution, right? Now, if we think about performing this very same experiment in a situation where we have noise in the evolution, think for instance of having PUD phasing, then the amplitude of these oscillations will be reduced, and after some time it may be that we are holding just a purely mixed state with no coherence whatsoever between the excitonic states. Okay. So here we can already identify what do we mean for long versus short-lived coherence. Superpositions in monomers involving only the ground and the excited state are done in a few femtoseconds. However, oscillatory signals that were assigned to excitonic superpositions were considerably longer lived, even extended towards the picosecond domain that, remember, is the typical time scale covering energy transfer in FMO. So of course, the situation in reality is more complex, right? The type of nonlinear spectroscopy, nonlinear means nonlinear in the number of pulses, involves in the generalization, if you want, of pump probe experiments, involves two pump femtosecond lasers with a separation tau, the coherence time, right? Then we let the system evolve for a waiting time, capital T, and then we'll have a detection time controllable, right? Okay, the point is that given that we have these time separations between probe, this variable detection time T, then it's possible to actually make Fourier transformations of those time variables in such a way that we can present the spectrogram in a 2D form in such a way that we can make a correlation map in between the excitation frequencies and the detection ones, right? So if there is coupling in between the pigments, right, then what can happen is that there can be in our spectrograms features where energy is absorbed and emitted at different frequencies. So this is a signature of coupling and allows, among other things, detect in which way energy is being transferred across the complex. Maybe just that you get a picture of what type of information this spectroscopy allows you to get. If you then think about varying the time in between the two pump pulses and the detection one, and focus your attention on one of the features, for instance, one of the cross peaks, if you follow what is the evolution of a selecting regions as a function of the waiting time, then oscillatory signals come out, right, which in particular may involve an oscillation at the frequency gap of the exciton separation. So then the initial observations, if you want, is that there are oscillatory features in the 2D signal from different complex, not also FMO, but also reaction center and the units present in certain type of algae, lead then to suggesting, you see that I'm being conservative with the wording, right, that given that this coherence can be observed on times that are relevant for something important to happen, in this case, excitation energy transfer, there could be a potential relation between the presence of that coherence and biological functionality. So this is what we want to scrutinize. Let's say that we will try to see first, how is it possible that can happen? So is it the case that we can really see that indeed long-lived oscillations could be observed? Second, what is the microscopic underpinning? What originates them? And then finally, see whether we can actually link that with some connection with actual biology. Okay? So there is already even a back of the envelope calculation that you could do, saying, well, actually, I'm not that surprised that there could be a difference in between the lifetime of oscillations involving ground excited versus excitonic ones, right? Because here, forget about the 800, 850, this I wrote for a different complex, but think about the general Hamiltonian where you have side energies and then an excitonic coupling, right? And now I diagonalize it coming with my excitonic states, right? If initially I have a given dephasing rate for the uncoupled systems, right? If I now make the admittedly far-reaching assumption that if I would be able to describe the dephasing of an isolated chromophores via a master equation with certain rates, right? If I make the assumption that the dephasing of the excitons will be ruled by a master equation of the same functional form, now with different coefficients, well, a back of the envelope calculation tells you that as a result of the delocalization due to this coupling, then you can have an effective reduction of the dephasing for the excitonic superpositions. So that could be, you know, a guiding idea that maybe as a result of the coupling, that's not impossible to happen, right? But take into account that we would like to even go further, right? So we want not only being able to explain how is it possible that there could be significant coherence between excitonic states in the presence of a possibly very aggressive vibronical environment, but we would also like to say this coherence should be able to facilitate something. Very much in the spirit, you will conclude the story saying, well, very much like I have 100% fidelity to perform qubit teleportation if I share an EBIT of entanglement, we would like to say something like, okay, this process will happen with this or that efficiency if and only if there is coherence in the system. So this is something tricky, very nice, and still to be done, right? But for the moment, I think we can understand what is going on microscopically for possibly having long-lived coherence in the systems. Right. For us, given that I adopted the historical perspective, let's say that when we heard about the experiments of Fleming and his group, we were at that time really trying to understand under which circumstances was advantageous to have noise in the system to perform information processing. And we knew at that time that, in fact, if we wanted to have steady state entanglement, right, it was advantageous for the system to be open. This is something that is now mainstream, right, but in 2002 was still a little bit of an exotic result. So somehow we have the hope that maybe in these complexes, something like that, a productive collaboration between coherent and incoherent interactions would be the reason why we could have this long-lived coherence. Okay, so what would be your starting point if you have a background in quantum optics? You will say, well, the first thing I will try to do is really divide the universe into two pieces, identify my system of interest with the network of chromophores, dipolarly coupled, and I put in this blob in orange everything else, all the vibrations in the world, okay? And I make the assumption, right, that the noisy processes that will be affecting these coherent dynamics will be a dissipation term that will make excitons relax and also some residual pure dephasing. So let's see how far we can go with this very simple model. Right, so we identify as our figure of merit what would be the amount of energy that is being transferred to the reaction center, modeling this transfer process as an incoherent transfer from the lowest lying exciton into the reaction center. Then what we obtain is that if you would have a network that doesn't exhibit any dephasing, so if in the case that I do a simulation in the side basis, take into account that the only noise present would be pure dephasing, right? The information we have is that physically the source of noise is actually local per chromophore. There doesn't seem to be signatures from molecular dynamics of spatial correlations. So this is a calculation with a master equation formulated in the side basis and computing what would be the excitation probability in the reaction center. So we are measuring our ability to efficiently convey energy as a function of the dephasing rate. So if the system is not noisy, this network will be highly inefficient. You will tend to delocalize the excitation, right, in such a way that some of the delocalized eigenstates don't have any overlap with the pigment with the lowest energy, or not the lowest energy, the closest to the reaction center. And as a result, roughly only 50% of the excitation will reach it. Something I didn't emphasize is that there are no signatures of having more than one excitation at a time, right? So we have from the beginning the simplifications that we can do the discussion in the one excitation sector within this picosecond time scale. Should you increase what is the level of dephasing, what you encounter is that actually it's possible for certain optimal dephasing rate to reach 100% efficiency. So perhaps not that unsurprising nowadays. What is clear is that we need a combination of coherent and incoherent processes at play in excitation energy transfer in such a way that there is one physical mechanism that allows for unfavorable configurations to emerge. So the behavior that we encounter if we plot what is the probability to convey energy to the reaction center as a function of the defacing rate is this typical non-monotonic curve where we have an increase in the transfer, right? If we modify, upgrade the defacing, right? Until reaching a region of optimality, and then should you continue increasing the noise rate, then you will effectively localize the excitation in a given site. So there were independent results at the same time by the group of Alana Spuru, and now this type of phenomena is normally referred to as NAQT, so environment assisting excitation transfer. I think for quantum opticians, it's easy to understand that indeed that's something that can happen, right? And a very nice textbook demonstration happened a few years ago in the group of Rainer Blatt and Christian Brott, where they consider an array of, I think they were like, okay, a 10-qubit network, and you see how they could reproduce experimentally nicely this behavior of the excitation transfer as a function of the defacing rate. So that was, let's say, the first. We can still keep even within the back of the envelope calculations. The first results pointing towards the fact of having a combination of coherence and incoherence at play in this type of complexes. The validity of the type of master equations that we have used, should they be obtained ab initio, you know that will rely typically in the validity of von Markov approximations, right? So having really being in the presence of a reservoir that should be as close as possible as delta correlated. If we look at what is the information we have concerning the spectrum of the fluctuations of the phonon bath in these complexes, you would be a little bit stressed to see what happens, and is that the environment is very structured, right? Moreover, the presence of underdamped modes that can be many, despite the coupling to each of them individually may be small, given that we have many and they can extend over large values or high frequencies, makes really the typical assumptions for the validity of reduction descriptions sitting on the fence, right? So we need, if we want to make any connection to actual photosynthetic complexes, we need to move forward and being able to predict what sort of behavior should we expect when we include the complexity of the phonon bath, right? And moreover, take into account that reading out properties concerning the specific details of the transfer from spectroscopy is far from trivial. So for our theoretical apparatus to be predictive, we need to account for the spectroscopy as well. Right. So then the next work we did was trying to incorporate some structure into the bath and take advantage of the type of numerical methods that have been developed to tackle many body physics in a numerically exact manner. So I'm just controlling how I'm doing with the time. So let me present you the first analysis of this type, where actually consider a dimeric system, right, with a type of parameters that were inspired by the first and second chromophore in FMO, that are the ones that are strongly coupled. Right, so if we write down what would be the Hamiltonian, I'm using a parameterization where I will couple to the displacement of a harmonic oscillator path in a way where typically you will write the complete constant if you want to compare it with the physical chemistry literature in terms of what they call the Juan-Riesz factor, Sk, right? Then we have that G is equal to square root of the Juan-Riesz times the frequency. Okay, so one way to get intuition about what can happen in the presence of a dimeric system that is dipolarly coupled by this interaction and is also coupled locally to a phonon bath with environmental operator being the global displacement of these many modes, right? If you rewrite the Hamiltonian, right, in terms of excitonic states, then you see that the role of the environmental modes looks very much like a driving term that could then, should it have spectral features in the vicinity of excitonic gaps, can act as an element that may regenerate the coherence that is lost as a result of the effect of all the other vibrational modes that are far from resonance. So in a very cartoon manner, think about of an spectral density, right, that exhibits a very sharp spectral peak due to the presence of an underdamped vibrational mode in the vicinity of the excitonic transition, right? So if we prepare an excitonic superposition, remember the effect of a first Ramsey pulse in an interferometer, right? Then should the evolution be unitary, this will simply precess at the excitonic frequency, but in the presence of local dephasing in the exciton bases, we will now be thermal relaxation of excitons. This will try to decay, right? However, if we have a mode that is close to resonance, we can then imagine the action of this mode as really trying to drive this transition coherently and competing with the relaxation component of the rest of the modes on the excitons, right? So we could expect that indeed the presence of these underdamped modes in the vicinity of the resonance may allow the beating to persist longer. You can say, okay, that's all blah, blah and cartoons. So what did you obtain? Again, we wanted to escape from reduced formulations. So from formulating reduced master equations for the excitons, right? In order not to be ransom for all the unavoidable approximations that one would have to make, right? In a situation where the microscopics is still relatively not as well characterized as in a quantum optical scenario. So for that, what we did is taking advantage of methods that implement density matrix renormalization techniques, right? And use an efficient procedure to actually map out our dimeric system coupled to this complex phononic spectral density, right? Made up of one that has an experimental ground, right? And then combined with essentially a very sharp Lorentzian peak. The important thing is that it's possible to unitarily transform now this initial configuration into a 1D configuration where the coupling between the new effective modes is only happening at the level of nearest neighbors. And this is an architecture that is friendly for a simulation with the energy. So then the results that we can obtain are numerically exact. And this is what we get for what would be, should it be accessible experimentally, right? This is a fully theoretical analysis. We compute the evolution of the whole world, right? Electrons and photons, right? And then we trace out the phononic path and examine what are the properties of the excitonic density matrix, where you can see that indeed, in the presence of an underdamped quasi-resonant mode, we are able to sustain the oscillatory behavior. These were simulations done at Lottie 77, but the interesting thing is that we can also show the persistence of oscillatory behavior also at room temperature. So then this analysis suggested that actually the key word may be here the formation of bibronic coherence as a result of having the possibility to have coherence at two levels, one that results from the excitonic degrees of freedom, and the second one from the interaction of those with selected vibrations. So the model was actually successful in describing experiments with artificial systems. And these are two references where specifically the theory was used. And in parallel, let's say that the same type of theoretical framework was also used to analyze the now not energy, but actually a process that may even be more relevant from the biological point of view, that is charge separation in the reaction center. So both the group of Rick Van Grondele and the group of Jennifer Ogilvy observe the observation we have for the efficiency of the charge separation were consistent with the identification of modes in the vicinity of the resonance of the special pair. Right, so you may see reasons to believe that indeed there could be long-lived coherence in pigment protein complexes. However, there is not yet a complete agreement, not in the presence of oscillations that I think everybody believes, and I think you only need to enter into a laboratory for these spectroscopies to realize that there are oscillations everywhere. But the issue is to which exact mechanism they are attributed. So in particular, this is a penis, but actually I may not have included right now. There is even a more recent reference in Science Advance led by Dwayne Miller, where they argue strongly on the purely vibrational origin of the oscillations, saying purely excitonic coherence will be very, very short-lived, and really what we are observing is mainly oscillations that are due to contributions from the ground state. So oscillations between the ground state and the first phonon, which obviously has the same splitting as the exciton one. Okay, so let's try to contest that and arguing that no, that may not be the end of the story, because take into account that we started including a little bit of the complexity of the phonon bath, but far from really addressing what is going on with the true spectral densities. So then these are results that are currently under revision, and you see that there is a bit of a gap in between the results I just explained, right, and the ones I'm going to cover in the last 15 minutes. And it's really due to the fact that we didn't have tools to tackle what is going on if we want to include all those modes that are happening in the high frequency region. So what we've done now is took a while to really be in a position to say, okay, there are 55 underdamped modes with a line width of about 1 picosecond. And we want now to be able to include them all in the simulations and even being able to move to room temperature. So, and we want to compute spectroscopy. We may still do the calculation of propagate everything, trace out phonon, see how does the excitonic density metric look like, but also we want to be able to compute theoretically what is actually measured. And those are spectroscopy being linear or multidimensional. So we consider then two model systems, which are truly dimeric actually. One is the water-soluble chlorophyll protein, which is another very well-studied complex, irrelevant biologically, but a nice toy model for theoretical studies. And another one is a very relevant system for biology because it's the special pair in bacterial reaction center. So if you ask biologists, they think that if any optimization is happening, it's much likely to happen for charge separation than for energy transfer. Okay, so the point is that if we look at the spectral densities, we are considering one dimeric system where the splitting is far away from the high energy tail with many underdamped modes versus the reaction center whose energy splitting falls in the middle. So what do I mean with this energy splitting? This would be the one that is computed to fit linear absorption in the case where the spectral densities are coarse-grained. So this is a value that has been then adjusted in such a way that with this spectral density and that gap, we reproduce linear spectra. So like the table I showed you at the beginning. Okay, so actually, to understand what is going on, we can even do simple perturbation theory to understand what sort of renormalization effects will happen as a result of the presence of these many modes. What we obtain is that the splitting will change, very much like lamp shifts are happening in quantum optics, and the new effective splitting that will concern now bibaronic states, right, can actually be smaller than the previously estimated or larger. What is the significance of this? Well, the significance of this is that the way to determine what are the values of the couplings I introduce in the system Hamiltonian are derived from here. And if this is different from that, and if this is different from that, then what happens is that the new bare electronic couplings that I will consider now are different from those initially. In particular, the electronic interaction can actually be larger. And this is very significantly for the interpretation of the oscillatory features in 2D. Because one of the standard arguments is that the interaction between pigments is not strong enough. But the point is that there is a very large uncertainty in what that value can be. In particular, can be significantly larger. So definitely the issue of ascribing oscillations to being purely vibrational or being of vibronic origin, I would argue that is still to be confirmed. But definitely there are reasons to believe that when we do a full analysis, including the complete spectral density, we cannot just simply smooth out this part without paying a very serious price. So just to illustrate how powerful the calculations can be, these are actually results for exact absorption spectrum, right? That, I mean, even, let's say, to be more convincing, we use not only our second generation TEDOPA, but in fact, we use hierarchical equations of motion to be able to have a different numerically exact technique, right? And what we can see is that in green, you have actually the spectrum that has been measured, right? What we obtain is that it's possible, if you look at the diagram we have in C, we can fix it completely, right? You see that the agreement is really good with the curve in black. When we include the 55 modes, and should we start coarse graining, you see that if I now consider 40 instead of 55 or 20 instead of 40, you see that the distance and the shape in between the two main absorption peaks is modified. So in fact, should I repeat the calculation of the absorption spectra exactly with the initial estimation for the excitonic gap, I get it totally wrong. So the effect of this very complex environment is absolutely crucial to be able to account for the spectroscopy. And we did a similar analysis for the special pair in the reaction center, where actually the effects as a result of having a larger electronic coupling are even more starking, right? And then we can see, when analyzing both signals in the cross peak of the actual spectral response, or doing the analysis of the properties of the excitonic density matrix, that we do observe oscillations in the excited state manifold, which have an electronic component and that live on a picosecond time scale. So by means of using numerically exact techniques that can now include the full complexity of the spectral density, we argue that the contributions of the purely excitonic coherence to the global 2D response may have been underestimated. So the debate is still on right? And one of the take-home messages of the talk is that in this type of complexes, connecting dynamics and a spectral response is not a trivial matter, in the sense that you are building up modeling, right, relying on parameters whose estimation is model dependent, okay? So therefore, in order to unambiguously link an observation, for instance, into the spectroscopy with an underpinning dynamics, right, then we need to be able to have methods that allows us to simulate the actual spectrum of the fluctuations. Right, so even if you would believe that what I said so far makes sense and that pigment protein complexes seems to be a nice example of a composite interacting system exhibiting long-lived bibronic coherence, we still have pending the big question of saying whether this coherence is actually the responsible for some enhanced function of any form. And this is still pending to be answered satisfactorily, but in my opinion, the framework of thermodynamics is probably one that is particularly well suited to answer whether there is any connection between coherence and function. So just very briefly, ultra briefly, let me just tell you some initial work we did in that direction, that is thinking about the pigment protein complex as a thermal machine operating in between two baths, right? The thermal bath from which they extract excitation and the phononic one where they dump the excess energy. Well, what is interesting to see is that depending on whether or not you include localized vibrations, right, you can have a performance of your thermal machine, right, that exhibits larger or smaller efficiency depending on whether the localized modes are present. In particular, the energy, we can define an associated power, so energy population per unit time, right, in such a way that we compare now the power delivered for complexes where I will have, let's say, a smooth spectral density versus one that includes localized modes. Actually, the power in the coherent case is enhanced. So let's say to link function and coherence in the context of biology, we will need something like that, being able to identify specific tasks whose performance is unambiguously enhanced by coherence, or facilitated or made possible at all. So that would be the loop we would like to close, right? So far, you've seen that in the light of the latest result, I would argue that the analysis is quite solid in here. The issue of identifying an unambiguous relation between enhanced function and coherence, we have some indications. So the view of having these cooperative effects between noisy and coherent dynamics seems to be suitable to think about these systems. And even if we forget completely about actual natural complexes, I always like to show this slide telling that what is the answer to are quantum effects relevant for biology? Maybe not, right? But very much like flying objects, right, do fly and they don't do it like birds, we can exploit biology to really identify basic design principles that can be harnessed in artificial scenarios, right? And I think don't have time really to tell you an example, but very much inspired by now large membranes of photosynthetic complexes. We have a couple of nice examples recently. So if you are interested, I can give you the exact references. So let me just finishing with some comments on what is really in the pipeline to move forward. So we expect that soon there will be the next generation of experiments involving both natural but also artificial complexes that have a degree of controllability. People are trying to develop at least theory. There's already quite a few papers trying to use entangled light sources to analyze what would be then the response of PBCs, including control that would be able to address specific regions in the two-dimensional spectroscopy. Recently, the group of Fleming has developed now mixed electronic vibrational spectroscopy to track simultaneously also the nuclear degrees of freedom. We have the aim to move beyond dimeric system and include in the full spectral density to be possibly able to determine to the response of ethanol. And also from the very theoretical point of view, it would be nice also in trying to establish this relation between coherence and function to unambiguously characterize what are the sets of operations that can or cannot achieve a given task given some resources. There's been some work by Nicole Junger group and by us in the specific case of photoisomerization, right? And arguing what type of underlying evolution would be able to exhibit higher or smaller efficiency. Many people that contributed all over these years to the efforts in the group I've just highlighted, right, the people that are currently here, Martin and Jeremy, and you may know that Felipe tragically passed away in an accident in the Alps. So some review articles in case you are interested in going further, and also I've been very endogamic, mainly commenting on our results. There are of course many others by other people in order to construct what seems to be coming out, that this is this bibronically coherent PBC. Okay, many thanks for your attention. I hope I didn't overshoot too much. Thank you.
Thank you, Susana. Now we have some time for questions. There was already one in the chat box. You can also read, but I can read it for you. This is about a porphyrin alignment and distance. Is known to change energy level alignment without changing the color. There is a reference, and the question is there a structure-related effect in these protein complexes? That is, how the iron oxidation is controlled at the porphyrin in hemoglobin.
Right. Okay. Take into account that here we don't really enter at this level of chemical detail, right? So for me, let's say, what is really, from the point of view of chemical groups, underpinning the behavior of the protein itself, right? So what is really, so this is the chromophore, and you talk about what may be happening with the residues of the protein in the vicinity, right? So for me, all that detail is encapsulated in the spectrum of the fluctuations. So what is, if you want, the microscopics behind that is something that at this level doesn't enter.
this was an answer for our colleague. Okay, thanks, Barış.
Maybe just one comment for Barış.
Okay.
I think this comment actually emphasizes something, and is that to really make progress, right, we need a combination of studies, right? So let's say we can maybe shed light in some directions, right? But for instance, the type of question you put forward, right, that would come really from the physical chemistry community is also essential in being able to characterize in the more precise manner, right, what sort of description of the bad we can consider as realistic. So unless there is crosstalk in between communities, I think, especially in the long-reaching aim of making some relation to actual biology, will not happen. Okay? So there is a bit of talk, but definitely there should be more.
Okay. Actually, I have a question, Susana. This, with your experience in this vibronic coherence, what is your view about this explanation of lithium experiments, lithium isotope experiments? There was an initial claim about this role of spin, quantum origin, but there is also an alternative description where you can consider the different masses of the isotopes of lithium. Do you think that is it something related to vibronic coherence? Do you have?
To be honest, I don't know. This is something I've not studied, right? And I cannot tell you. It would be too risky to say anything. So okay. I don't know. Maybe, maybe not.
And something about this, what you call this noise assistant transport. Is there I mean, can you make an analogy with so-called resonant tunneling? You know, this was an interesting problem in the 80s after SI-Berg.
Yeah.
Is it something like opening a hole and then enhancing?
It really indeed belongs to the same category, right? Of having, let's say, the possibility of breaking coherent superpositions by means of incoherent effects, right? So in our case, if you think about forming the localized excitons that will not overlap with the reaction center, the situation of having dephasing and enhancing the transfer is very much like having a Mach center interferometer, right? Where you never, if it's perfectly balanced, right? For having no clicks in one of the detectors to include noise and now having clicks in both. So indeed, I would argue that it's not a surprise at all that the presence of noise will actually be favorable for transport. And indeed, there are many, many instances where it happens elsewhere. The issue is whether it's interesting that happens precisely in this type of complexes, right?
Yeah, I see. Okay, thank you. There is one more question. I think we have some time for this from Barış again. If you want, you can read using simplest Guterman symmetry cascade argument. Each porphyrin is 4 oscillators in one, with each oscillator having a different dynamic correlation coefficient. Combining this with the flexibility of porphyrins, there is a rich chemistry. Did anyone
investigating this? I cannot tell you really. With this, you will have to really talk to the chemist. So, for instance, one person you could talk to is Elisabetta Collini in Padova. Elisabetta did very pioneering experiments with complex in algae, and she's a chemist by training, right? And definitely has tried to investigate microscopically the solvent interactions. And this would be a person I would target for this type of questions.
Okay, thank you very much. Is there a question?
Onur.
Yes. Can I ask my question?
Okay, please.
Zafer Hocam. I have a question regarding the utilization of resource theoretical approaches in this problem of photosynthesis. As far as I know, it's not too easy to generalize thermomodularization criteria in the cases where you have some coherences in the energy basis.
It's not easy what? Sorry, I didn't understand you.
As far as I know, it is not easy to generalize thermomodularization criteria if we have some coherences in the energy basis.
Okay, now you're talking about the case of employing resource theoretical.
Yes.
Yeah.
Yeah.
Okay, that I think it will be very incomprehensible for everybody in the audience if you has not read the papers. But what you are saying is absolutely right. That's why in this type of resource theoretic approaches, what you tell apart is certain type of operations cannot do. So let's say in our specific case, right, where we analyze the process of photoisomerization, what we consider are the abilities of operations that are not only thermal, but locally thermal, in order to be able to define in a consistent manner what would we mean by Markovian thermal operations. So if you ask me what happens if I remove, and there the conclusion is that non-Markovianity is able to enhance the performance, right? But there is a caveat, because you could be able to break the locality assumption. You could have operations that are globally thermal, but not locally thermal, right? That could be Markovian and maybe have a larger efficiency. And indeed, the reason I cannot solve that question right now is because I wouldn't know how to use thermomodularization if I break local thermality. So that's a very good question.
Thank you very much.
Would be nice to do. Yeah.
Thank you very much. Thanks. Thank you, Susana. We thank our speaker once again.
Thank you. Thanks a lot.
Thank you.